Hello dear students, I am Namrita. I will be teaching you biology with respect to second PUC, CET and NEET. Okay. So before getting into today's topic, I will just give you some quick tips, okay, which will be very helpful for you to prepare for your entrance exam, okay, to crack it very easily. Okay, just don't get panic whenever you assume that this is an entrance exam. It is just an entrance exam. Okay, you should feel free while learning or also preparing. Okay, so first of all, you have to just have an idea that I can do it. Always have a positive mindset. That is a stepping stone for your success, right? So concentrate on whatever you learn. Make your own notes. Focus on whatever you learn. Okay, you will definitely crack it. Okay, it is not a big hailstone. It is really very easy when you feel it. Fine. So with that positive vibes, let us start with today's chapter. So today we will be starting freshly our journey of KCET or NEET entrance exam preparation. Alright, are you all ready? Yes. So firstly, we will be learning the first chapter of your biology, that would be sexual reproduction in flowering plants. Hope you all know the first chapter of previous, which is previously to this was reproduction in organisms, correct? That is not there. So this will be your first chapter, okay, which is that sexual reproduction in flowering plants. Okay, now let us do some introduction to this chapter. Okay, sexual reproduction in flowering plants. See, this chapter itself will give you many meanings or definitions as in it. See here, before moving on to the entire chapter, we will just have an overview on what and all we will be studying in this chapter, right? So, as we look into this chapter name, the first word which hits our mind is reproduction. Reproduction. So, what is reproduction? That you have learned since grade, 6th, 7th, 8th. You've been learning reproduction only, right? So, what is reproduction? What do you understand by the term reproduction? Reproduction is, in simple, said to be, it is a biological process. What is reproduction? It is a biological process which happens inside a body, especially female body, right? Reproduction happens in a female body wherein there will be fusion of male and female gamete in order to produce new offsprings. In order to produce new offsprings is what we understand by the term reproduction. Correct? Reproduction. Again, in that we have two types. What are the two types of reproduction? What are the two types of reproduction? It is asexual reproduction and sexual reproduction. Okay, these are the two types of reproduction we have learnt so far. Now tell me, what is asexual reproduction? Asexual means that is not sexual. Before that, we will learn what is sexual reproduction. Okay, what did we define now in reproduction? In reproduction is a biological process which will give rise to new offsprings, correct? So in the same way, sexual reproduction means, Andre, here there will be involvement of two parents here. But in asexual, only one parent is required, only one parent. That is the simple difference between asexual and sexual reproduction. Okay, in sexual reproduction, what happens? There will be two parents, male and female parent. where both parents will produce gametes, male gamete and female gamete, and those two will fuse together. Then the produced offspring is said to be from the sexual reproduction. But here what happens, in asexual reproduction, there will be only one parent from which the new offspring is being developed. That is through various examples or through various types like budding, fragmentation. See, all these vegetative propagation, all these are examples for asexual reproduction, right? For sexual reproduction, all the mammals we see, all the mammals or animals will, or plants as well, because we are focusing more on sexual reproduction in plants today. So we see sexual reproduction in majority of organisms in the world. Fine. So this asexual reproduction can be also done artificially and then man-made. That means what? So man can cut and graft. Okay, you can cut and graft. Or you have also seen in your gardens. You also have garden, I think. So you will also do plantations. You will just take out the stem of one of the plant and just go put it into the soil so that another plant will generate. That is also one type of artificial asexual reproduction, correct? So these are all types of asexual reproduction. Fine, after learning the types of asexual and asexual reproduction, we have been talking about reproduction many times. Now tell me, why reproduction is essential? Why do you think so? Reproduction is very important. Yeah, okay, reproduction process is very important because reproduction ensures, okay, it will ensure to keep the particular species to continue its generation. Okay, the continuity of the species from one generation to another generation is happening because of the process of reproduction. For the continuity of the species generation, reproduction is so important, correct? Vamsha beliyad hege. In case of humans, like that. Or to preserve one kind of species, it should give rise to another new species and die. Or else the particular species will just go extinct. Adik iralla. Next ಇನ್ನೊಂದು 10 ವರ್ಷಕ್ಕೆ ಇರಲ್ಲ. ಅದೇ ಆ ಸ್ಪೀಶೀಸ್ when it gives rise to new individuals ಏನಾಗುತ್ತೆ, ಇನ್ನ ನಮಗೆ ಮುಂದಿನ ಫ್ಯೂಚರ್ ಜನರೇಷನ್ಸ್ ಅಲ್ಲಿ ಕೂಡ ಇರುತ್ತೆ. So this is also one of the importance of reproductions. Got it? Understood? So after learning some of the introduction, again I will get back to the chapter name. I will tell you why. See, we have learned reproduction andre ಏನು ಗೊತ್ತಾಯ್ತು, sexual reproduction andre ಏನು ಗೊತ್ತಾಯ್ತು. Now in this chapter we are focusing on flowering plants. Now what is so special about flowering plants? Why are we learning that? Why are we learning about flowering plants now? Yes? Why do you think so? The classification of plants, right from the algae, we have algae, bryophytes, pteridophytes, gymnosperms, and then comes the angiosperms. See, these angiosperms are, the highlight of this class. What are angiosperms? You've already learned, I think, in your previous classes. Angiosperms are nothing but flowering plants. What are they? They are nothing but flowering plants. So, what is today's our topic of interest? We are learning about sexual reproduction in flowering plants. Got it? So, flowering plants are also called as angiosperms from the classification of plants. So, the angiosperms have been evolved due to evolution of gymnosperms. They have been evolved from gymnosperms. Gymnosperms in the evolution ಆಗಿ angiosperms ಆಗಿರುತ್ತೆ. ಹಾಗೆ algae in the bryophytes in the pteridophytes in the gymnosperms, and then to flowering plants. These are the complex level of plants. Got it? So today we are learning sexual reproduction in flowering plants, also called as angiosperms. Fine. Yes. So in flowering, okay, before going to the exact topic, plant structure, I think you all remember plant structure. Yes or no? See, plant structure, how is it? It is having a root system, bottom root system. Then above the soil you have shoot system, which gives rise to stem, and stem bears many leaves in there. We also have this beautiful, fascinating structure called as flower. Correct? This is the basic structure of a plant. I've just drawn in a just a rough manner, not exactly a clear image, just to make you understand. That's all. Fine. So here we understand that what is the work of the roots? To absorb the essential nutrients, correct? Absorb essential nutrients and water from the soil. Then what is it? A function of stem here? Why stem is there? Stem function is to conduct whatever absorbed by the roots. Ivaga roots absorb ಮಾಡಿರುತ್ತೆ, ಕರೆಕ್ಟಾ? ಎಲ್ಲ ಬೇಕಾಗಿರೋ ಅಂಶಗಳನ್ನ ಸಾಯಿಲ್ ಇಂದ ಅಬ್ಸರ್ವ್ ಮಾಡಿರುತ್ತೆ. ಅದು ಟ್ರಾವೆಲ್ ಆಗ್ಬೇಕಲ್ಲ, ಇಡೀ ಪ್ಲಾಂಟ್ ಯೂಟಿಲೈಸ್ ಆದ್ರೆ ಅದ್ರದ್ದು ಹೆಲ್ಪ್ಫುಲ್ ಆಗುತ್ತೆ ರೂಟ್, ಇಲ್ಲಾಂದ್ರೆ ಏನದು ಹೆಲ್ಪ್ಫುಲ್? ಈಗ ಇದು ಕಂಡಕ್ಷನ್ ಮಾಡುತ್ತೆ ಸ್ಟೆಮ್. ಓಕೆ, ಕಂಡಕ್ಷನ್ ಅಂದ್ರೆ ಏನು? ಯು ಹ್ಯಾವ್ ಆಲ್ರೆಡಿ ಲರ್ನ್ಡ್ ಅಬೌಟ್ ವ್ಯಾಸ್ಕ್ಯುಲರ್ ಟಿಶ್ಯೂಸ್, ಇಸ್ಂಟ್ ಇಟ್? ವ್ಯಾಸ್ಕ್ಯುಲರ್ ಟಿಶ್ಯೂಸ್ ಅಂದ್ರೆ ಏನು? ಝೈಲಮ್ ಅಂಡ್ ಫ್ಲೋಯಮ್. ಝೈಲಮ್ ಅಂದ್ರೆ ವಾಟರ್ ಕಂಡಕ್ಟಿಂಗ್ ಟಿಶ್ಯೂ, ಅದು ಫ್ಲೋಯಮ್ ಅಂದ್ರೆ ಫುಡ್ ಕಂಡಕ್ಟಿಂಗ್ ಟಿಶ್ಯೂ. ಅದೆಲ್ಲ ಸ್ಟೆಮ್ ಅಲ್ಲಿ ಇರುತ್ತೆ. ಸೊ ವಾಟ್ ಎವರ್ ಅಬ್ಸಾರ್ಬ್ಡ್ ಇನ್ ದ ಸಾಯಿಲ್ ವಿಲ್ ಬಿ ಟ್ರಾವೆಲ್ ಥ್ರೂ ಔಟ್ ದ ಪ್ಲಾಂಟ್ through this conducting system called as xylem and phloem in the stem. Correct? So it will travel every part of the plant. And again, you also know leaf has a major function in here, that is photosynthesis. It will prepare its own food. Correct? It will prepare its own food, and again food should be traveled to all the parts. That is done by phloem. Correct? This shoot has given rise to this beautiful, different, unique structure called as flower anta gottaಯ್ತು ನಮಗೆ. See, the shoot is being only modified to give a beautiful, colorful, unique, large, with some fascinating smell, aroma. Correct? So that is flower, and it's very colorful, unique. It is different in different species. Correct? It varies in shape, size, color, texture, number of petals. Everything will differ according to the species. So, ivaga namge onondu part of the plant has specific function anta gottaytu namge. Root ಗೊಂದು function ಇದೆ, absorption. ಈ shoot ಗೊಂದು function ಇದೆ, conduction. Leaf ಗೊಂದು function ಇದೆ, food preparation. Why this flower is there? To only attract, to only give some color to the plant. Ashtena, the purpose. What is the purpose? Exact purpose of flower here? Of course, we learned that flower is a modified part of the shoot system in a plant. Correct? This flower, what is its necessity? Do you think only attraction? No. The major, actually the most important structure or organ in a plant is flower. Why? Because it is the reproductive organ. Avaglinda reproduction anta kaltkondi. Correct? Ee reproduction agbekanದ್ರೆ there should be some structure for it. Correct? What is that structure in a plant? That is flower. Flower is the most important reproductive organ in a plant. That is why that is the most important function of the flower, neither for attraction. Got it? So this flower, the structure of flower, the different structures present inside the flower and what are the function of the flower, we are going to learn in today's class. Got it? So let us move on. Let us move on to the chapter now. Yes. So we learned that all flowering plants shows sexual reproduction. Why? Why does it not show asexual reproduction? Why only sexual? If an question is asked in a CET, will plants produce through only sexual reproduction? Yes, yes or no, or both? What would be your answer be? The correct answer is both. Plants will be able to produce both from asexual as well as sexual reproduction. But mostly all the time sexual reproduction is the one which is being seen. Okay, this is in most cases. Most cases we see sexual reproduction in all the plants. Got it? Why it the flower only shows the sexual reproduction, or flower is a reproductive organ? Because it is having the gametes in there. What are the gametes? We'll discuss later. Okay, it should have male and female gametes. Sexual reproduction andre ನಾವು ಏನಂತ ತಿಳ್ಕೊಂಡ್ವಿ, sexual reproduction ಆಗಬೇಕು ಅಂದ್ರೆ there should be two gametes, male and female gamete. Then only sexual reproduction is seen. So what are these structures, male and female gamete? ಯಾವುದು ಈ flower ಅಲ್ಲಿ? We will learn it further. Okay. So before that, we are studying the reproduction. Correct. Reproduction is the process where it involves the fusion of male and female gamete and it will form an embryo. It will form an embryo. Correct. The studying of embryo, ಈ ಎಂಬ್ರಿಯೋನ ನಾವು ಸ್ಟಡಿ ಮಾಡಬೇಕು. ಕರೆಕ್ಟಾ? ಯಾಕಂದ್ರೆ ಅದೇ ನೆಕ್ಸ್ಟ್ ಹೊಸ ಪ್ಲಾಂಟ್ ಆಗಿ ಡೆವಲಪ್ ಆಗೋದು. That study is called as embryology. Embryology is nothing but it is a branch of botany, okay, which will explain on the nature and developmental process with the reproduction in angiosperms. Ashte. Okay, we are learning here the developmental process, its structures, its function in detail. Then that study is called as embryology. Easy. Embryo andre it is the newly formed individual. Correct? Logy andre study. Wherever you find the word logy, it is to study. You are studying embryo. So it is embryology. Got it? Yes. So see, we know that basic thing. ಇವಾಗ ನಾವು ಯಾವುದೇ ಅಥವಾ ಊರಿಗೆ ಹೋದ್ರು ಲೈಕ್ ಎಷ್ಟೊಂದು ಜನ ನಾವು ನೋಡಿರ್ತೀವಿ, ಒಂದು ಸೀಡ್ಸ್ ನಾವು we will just put above the soil and we'll water it and it will go some to few layers down. ಅವಾಗ ಏನಾಗುತ್ತೆ, it will grow into a plant. So ಆ seed ಅಲ್ಲಿ ಏನಿದೆ, seed from the seed only a small embryo will germinate. Ready? The small embryo, small plant. This small plant is called as embryo, which will turn into a new big individual. And this is the cotyledon. Cotyledon andre ivaga dicot, monocot antive. Andre the seed is having only one cotyledon. Then it is monocot. When the seed is having two cotyledons, it is called as dicot. So this is the cotyledon, the space, okay, the vacuole inside the seed, and the outer covering is seed coat. So what this image is showing you is this big tree is being grown from the small seed anta. It gives so many meaning. ಎಷ್ಟು ಅರ್ಥ ಬರುತ್ತೆ ಅಂದ್ರೆ, ನಾವ್ ಇವಾಗ ಯಾವುದೇ ಒಂದು ಕೆಲಸ ಮಾಡ್ಬೇಕು ಅಂದ್ರೆ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಈಗ ನಾನು CET ನೀಟ್ ಪ್ರಿಪೇರ್ ಆಗ್ಬೇಕು ಅಂತ ನನ್ನ ಮೈಂಡ್ ಫಿಕ್ಸ್ ಆಗಿದೆ, ಹ್ಯಾವ್ ದಟ್ ಡಿಟರ್ಮಿನೇಷನ್ ಇನ್ ಮೀ. ಅದಕ್ಕೆ ನಾನು ಓಕೆ ಜಸ್ಟ್ ಡಿಟರ್ಮೈನ್ ಮಾಡ್ಕೊಂಡು ಜಸ್ಟ್ ಫಿಕ್ಸ್ ಆದ್ರೆ ಸಾಕಾ? No, I have to keep one step further. One step further ಅಂದ್ರೆ ಏನು? I have to start getting prepared. ಏನೇನಿದೆ syllabus? ಏನ್ topics ಇದೆ? What are the reference books? What are my notes? From when I'll start? Okay, I've started with chapter 1. That is the growth of the seed. Seed ಇಲ್ಲಿ ಯಾವುದು? ನಮ್ಮ ಮೈಂಡ್ ಪ್ರಿಪೇರ್ ಆಗ್ತಾ ಇರೋದು. ಅದು ಎಷ್ಟು ಪ್ರಿಪೇರ್ ಆಗ್ತಾ ಇರುತ್ತೋ ಅಷ್ಟು ಬೆಳೆಯುತ್ತೆ, ದೊಡ್ಡ ಆಲ್ದ ಮರ ತರ. ಇಲ್ಲ ನಾವೇನು ಮೈಂಡ್ ಅಲ್ಲೇ ಇಟ್ಕೊಂಡ್ರೆ ಜಸ್ಟ್ ಯುವರ್ ಹ್ಯಾವಿಂಗ್ ಸೀಡ್, ಬಟ್ ಯು ಆರ್ ನಾಟ್ ಪುಟಿಂಗ್ ಎನಿ ಎಫರ್ಟ್ಸ್. ನೀವೇನು ಓದ್ತಾ ಇಲ್ಲ, ನೀವೇನು ಬರೀತಾ ಇಲ್ಲ, ಏನು ಪ್ರಿಪೇರ್ ಆಗ್ತಾ ಇಲ್ಲ, ಸೀಡ್ ಆಗೇ ಇರುತ್ತೆ. ಅದಕ್ಕೆ ಎಸೆನ್ಷಿಯಲ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್, ನೀರು, ಮಣ್ಣು, ಫುಡ್ ಎಲ್ಲ ಬಿದ್ದಾಗ್ಲೇ ಅದು ದೊಡ್ಡ ಮರ ಆಗೋದು. ಕರೆಕ್ಟಾ? ಹಾಗೆ ನೀವು ಕೂಡ ಪ್ರಿಪೇರ್ ಆಗ್ಬೇಕಾದ್ರೆ ಯು ಹ್ಯಾವ್ ಟು ಕೀಪ್ ಫುಲ್ ಆನ್ ಫೋಕಸ್. ಯು ವಿಲ್ ಅಚೀವ್ ಇಟ್. You will achieve it. Have that confidence, but not overconfidence. I'm telling you. Have that aim. You will really get to it. Okay. So this is the meaning of this picture over here. Okay. We will move on to the next slide, which says that the structure of the inflorescence. The inflorescence is a bunch of the flowers. Bunch of the flower buds or flowers is called as inflorescence. Right? The structure of inflorescence, flower, athva floral parts. Floral parts is the parts of the flower. It shows an amazing range of adaptations. Yake? To ensure the formation of the end products, that is fruits or seeds. A flower should become a fruit or a seed. Correct. So, the structure, adu hege structurized agide alva flower, its structure has some meaning. Okay, its texture has some meaning. That shows an clear adaptation for that flower to ensure easy process of reproduction. Okay, adapt ಹೇಗಾಗಿರುತ್ತೆ ಅಂದ್ರೆ ಅದಕ್ಕೆ ಯಾವುದೇ ಪ್ರಾಸೆಸ್, ಆ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ನಡೆಯುವಂತಹ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಪ್ರಾಸೆಸ್ ಆದಾಗ ಇಟ್ ವಿಲ್ ಬಿ ಏಬಲ್ ಟು ಫಾರ್ಮ್ ಇನ್ ಟು ಅ ಫೈನಲ್ ಎಂಡ್ ಪ್ರಾಡಕ್ಟ್ಸ್ ಇನ್ ಅ ಸೀಕ್ವೆನ್ಷಿಯಲ್ ಮ್ಯಾನರ್ ವಿತೌಟ್ ಎನಿ ಹಿಂಡ್ರೆನ್ಸ್ ಅಂತ ಅರ್ಥ. Got it? Yes. So flower, apart from the reproductive organ, it is also used for aesthetic or social or religious or cultural values. We can say that. ಹೇಗೆ ಅಂದ್ರೆ ಫ್ಲವರ್ಸ್ ನ ಆಸ್ ಅ ಸೈನ್ಸ್ ಸ್ಟೂಡೆಂಟ್ ಅಥವಾ ಬಯೋಲಜಿಸ್ಟ್ ಆಗಿ ನಾವೇನ್ ಅರ್ಥ ಮಾಡ್ಕೊಂತೀವಿ, ಇಟ್ ಇಸ್ ಅ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಆರ್ಗನ್, ಇಟ್ ಹ್ಯಾಸ್ ಸಮ್ ಯುನೀಕ್ ಫಂಕ್ಷನ್ ಅಂತ, or it is having its own purpose to serve anta. But for other common people, what they think, flower ಈಗ ಪ್ಲಾಂಟ್ ಏನಕ್ಕಾಗುತ್ತೆ, ತೆಗೆದು ಪೂಜೆ ಮಾಡಕ್ಕೆ, ದಿನ ಪೂಜೆ ಮಾಡ್ತಾರೆ, ಅದನ್ನ ಹೂ ಕಟ್ತಾರೆ, ಫಾರ್ ಆರ್ ಅದರ್ parties or other events, they will use it as some decorative purpose. Correct? So either aesthetic, social, religious or cultural values agi noಡ್ತೀವಿ. So apart from that, we know that flower is used as a reproductive organ. Correct? So as for a biologist, flower is said to be the modified shoot system. Not the shoot. This is the shoot. Correct? The shoot has been modified into this colorful flower. Correct? So to a biologist, we can define it as it is a modified condensed shoot. Why is it modified? Because it is having some function to do. What is the special function for it? For the sexual reproduction. Correct? The sexual reproduction. Why we call it as sexual reproduction? Because it is having both the parent gametes. Andre, for sexual reproduction, you know that there should be a male gamete as well as female gamete. Yeah, both these things are present in this flower. We will further learn what are the gametes, male and female gametes in the flower. Okay. So, flower is said to be modified condensed shoot to perform certain function, that is reproduction in the plant. Right? So, When we have to study the structure, the lateral section of a flower. Lateral andre, you just hold a flower and we will cut it from longitudinally and from top to bottom. When we cut down like that, we will visualize these structures. What are these structures now inside the flower? See, a typical angiospermic flower. Angiospermic, don't get confused. We already saw it in the classification. Correct? The most complex level of plants are called as angiosperms. So, angiospermic flower alli there important four whorls are there. Now, what are these important four whorls here? One is calyx. This is the outermost covering. Calyx. Yeah, calyx. So, calyx is sepal. Calyx is nothing but sepal. Okay, this is the first outermost whorl. Now, the outermost whorl. Okay, what do we call it as? Whorls. There are four whorls in a flower. Now, the outermost is called as calyx. Next, inside to it is corolla. Corolla is nothing but petals. What are corolla? They are petals. Okay. So, this is second whorl. Next, androecium. Androecium is said to be the third whorl. What is androecium? I can't find any androecium word over here. What do you think androecium is? It is nothing but the anther and filament, which is called a stamen. Correct? We've already learned in the previous grade. Since 5th standard, we've been learning this. Correct? Parts of a flower. So, anther and filament together is called a stamen. In stamen, we call it as androecium. This androecium is said to be the male organ. Male reproductive organ. And the last whorl is gynoecium. The fourth whorl, innermost whorl is gynoecium. Okay, gynoecium is this part. Yavadu? Ovary, style and stigma. Together, yenanta kariti vidna? Good. That is carpel. It is called as carpel. Alright. So, in a typical flower, we can find these four important whorls. What are they? First one, calyx, that is sepal. Second is corolla. Corolla and the petals. Third is androecium, the male reproductive organ. That is anther and filament together called stamen. And the fourth whorl is gynoecium. Gynoecium means the female reproductive organ. That is ovary, style and stigma. Together called as carpel. Now, did you understand the structure of a typical angiospermic flower? Okay, this is important for your board wise also, as well as they may be asking questions based on this in the CET or NEET. Okay, they will give this image and this give a blank space over there with the options. You have to give a correct labeling there. Alright? Yes. So, after this, after learning the typical structure of the flower, let us learn how these four whorls are divided. We said there are four whorls, correct? How are these divided actually? What are their functions? According to their functions, they are divided into two. What are they called as? Non-essential organs and essential organs. See, non-essential organs. What is the meaning of non-essential? Essential andre something is very important, which is needed anta. Essential, correct? Non-essential andre enagutte, which is less needed. Comparatively less needed. Ashta avashyakate illa anta. Okay, but it has some value, but not too much. Essential organs andre, these are very important organs which are very much needed. Now let us learn what are these words coming under non-essential and essential organs. Okay, now, so non-essential organs are also called as helping words that were accessory anta. They're just helping these essential whorls. Okay, na? They are also called as perianth members. Yakandre perianth, they are present in the outermost part of the phloem. Okay, so they are also called as sterile parts. Yakandre they are not directly involved in the reproductive function. That is why they are called as sterile. They do not have that capacity to reproduce. Only with these non-essential whorls, a flower cannot reproduce. Okay, but these are fertile parts, hence having capability for reproduction. Fine. So, what are these non-essential organs or whorls? Calyx. As we learnt in the previous slide, calyx andre eno? Sepals. Correct. And corolla petals. These two words will come under non-essential, accessory, helping words. Okay, calyx and the sepals, which are arising from the opposite sides of the thalamus or receptacle. Correct? The corolla or petals, they are also little colored and having different textures, shapes. Okay, and more attractive when compared to sepals. Sepals are usually green in color. Petals they are variable. Colors tumba irutte. Right? Now, what are these fertile essential organs? These are nothing but andrecium and gynecium. As we already said, andrecium andre male reproductive organ, gynecium andre female reproductive organ. So, andrecium is nothing but stamen. Stamen andre we already learned that anther and filament together is called as stamen. Carpels andre ovary, style and stigma. So, did you understand this classification? 4 whorls divided into 2 as non-essential and essential. Alright. So, with this, we will move further visualizing the animative view of this flower. I think you people can visualize here. See, there is this blinking. This part is called as ovary, the bulb-like structure. This is style. Right? And the topmost part is called as stigma. Fine? Ovary, style and stigma. This region, middle part is female reproductive organ. And these sides, you can see now, this filament and the anther, these are male reproductive organ. Fine.
Root 3 into root 3. When is the root 3?
With that, we will move further by learning in detail with these male and female reproductive organs. Okay, now when we have to understand reproductive process, first we have to understand what are the reproductive structures, organs what are the structure. Correct. So first we will understand these structures. Pre-fertilization before fertilization, we have to understand the structures involved for the fertilization process. And with that, moving on, we will understand the events or the process involved. Okay. So, in that, we are first going to learn about the male reproductive organ. Male reproductive organ. What are the structures involved in male reproductive organ now? Stamen. Stamen, by now you are all aware what is stamen, right? Stamen andre anther and filament together is called as stamen. Then microsporangium, this is new to you. This word is new to you, right? Microsporangium and pollen grain anta idu. First, Just don't get confused. Stamen anta talelilli first. Next, we will understand what is microsporangium and what is pollen grain. Okay, in detail. So, firstly, we are going to learn the male reproductive organ which is involved in the sexual reproduction. Ega, we know that this part, which is looking like a long, slender, narrow shape, very long, narrow, slender shape structure or stalk is called as filament. What is this called as? Filament. Okay. And what is the structure above the filament called as anther? You can see this umbrella shaped, correct? Either bilobed umbrella shaped structure that is called as anther. Okay, which is above this stalk called as filament, right? So, this is filament. Stamen has these two structures called as anther and filament. Anther is the bilobed structure. Either you can visualize this. See, there is one slit here which is appearing as two different divisions. So it is called as bilobed structure, that is anther. That is attached to some stalk here. That is called as filament. So stamen consists of two parts, that is anther and filament. Anther, as we saw, it is bilobed structure and also it is broad. Correct? Filament was very narrow, but anther was a broad structure. Correct? Broad knob-like structure, and this is said to be the fertile part. Fertile part. Which is the fertile part among filament and anther? Anther is said to be the fertile part. For having said that anther is the fertile part, we have to have particular reasons for it to prove. Correct? The anther na fertile part anta idhi. Filament na we are calling it sterile part. Alva? Why anther is called as a fertile part? Because it consists of the male gametes inside it. Male gametes are present inside it. Adikke adina navu fertile part anta karitivi. Filament, it is just an accessory part, it's a helping part to hold the anthers to the position. It is giving the mechanical support or strength to the anther. Got it? So, it is having two lobes. Anther is having two lobes. Okay? With the dissection in the middle, such an anther is called as diathecus. What is it called as? Diathecus. Yakandre, it is having two lobes. So, I will name, or scientifically we name it as diathecus. Alright. So, you can see here in this picture, this is the filament stalk-like structure, and above it you can see this bilobe. There are two cap-like structures which is stick together with the dehiscence and the one line separation there. So, this is called as bilobed anther. Correct? This is bilobed anther. This bilobed anther na, when we visualize under microscope, we don't naked eyes will cancel adu. When you take a section and do some process and observe it under microscope, then you can see this type of visualization. Yen kansite namge adana next further sectioning madadaga we can see these small two other sections in one lobe. See, there are two sections here. Nodi, in one section ಇದೆ. In one section ಇದೆ. In the same way, in other side also we have like this. There are four sectioning or group like that seen in each of the anther. Fine. So, in that there are again these small, tiny little spore-like structures. Okay. Don't worry about the naming. We'll further discuss on that. Just remember, when we dissect or section the part of the anther, we will visualize these 4 segments. Okay? And in that 4 segments, we can further see the small spore-like structures in it. Got it? What are those structures? Why is it present? We will further learn now. See, this is a typical flower. There is this filament and this is the anther. When we take sectioning, we will visualize in this form. See, more clearly we can see in this picture. Exactly the same radius it is having. See, it is not same in all the anthers. It is differing, okay? So you can see these structures. Now, what is this? 4 different segments called as. Each one, okay? Each one. See, there are 2 in one lobe. Another 2 in another lobe. So, together, each one is called as microsporangium. So, avalagale naven odidvi stamen, microsporangium, pollen sac or pollen grains anta odidvi alva. Now, that microsporangium ಏನು ಅಂದ್ರೆ ಇದು anther alli present ಇದೆ. Okay, it is present in anther. This is microsporangium. Again, in that we can find the small spore-like structure ಅಂತ ಹೇಳಿದೀನಿ. That is called as microspores. Clear? Microsporangium 4 in number, and inside the microsporangium we can find these microspores. Got it? So, a dithecous anther which is having 4 microsporangium, 2 in each lobe. The best example for that is Datura plant. Okay, this is the Datura plant picture. So, this is the one of the best example for a dithecous anther. Dithecous anther is nothing but what we visualized in the previous slide. Andre, it is having 2 lobes. Each lobe it is having 2 microsporangium, total 4 microsporangium together in a dithecous anther. Best example is Datura. See, either same thing we saw. But here we can see one more word, other synonyms. Okay, in biology synonyms are very important. By getting synonyms only, you will be halfly confused in your question paper. One day, very terms. You will think only one word and you will not learn other words, other synonyms, and you will get confused as you have not learned it. Okay, see, microsporangium anta karidvala, that is having another name. What is that called as? Pollen sac. Microsporangium ಏನಂತ ಕರೀತೀವಿ? Pollen sac ಅಂತ ಕೂಡ ಕರೀತೀವಿ. Pollen sac ಅಂತ ಕರಿಯಕ್ಕೆ ಪೋಲನ್ ಒಳಗಡೆ ಇರಬೇಕು. ಈಗ ಅಕ್ಕಿ ಚೀಲ ಅಂತ ಕರಿತೀವಿ, ಕರೆಕ್ಟಾ? ಅಕ್ಕಿ ಚೀಲ ಏನೇ ಕರಿತೀವಿ? ಅಕ್ಕಿ ಒಳಗಡೆ ಇರೋದಕ್ಕೆ ಅಕ್ಕಿ ಚೀಲ ಅಂತ ಕರಿತೀವಿ. ಅದೇ ತರ pollen sac ಅಂತ ಏನಕ್ಕೆ ಹೇಳ್ತಾ ಇದೀವಿ? Because it is having pollen grains. That is why we call it as pollen sac. See, consists of pollen grains. That is why we call it as pollen sac. These are other synonyms. Please remember, microsporangium. This word also you can split and learn. See, micro andre eno, small. Sporangium, spores andre eno. Spores. There are some tiny little structures. Sporangium. Angium is some scientific word. So, microsporangium. ಅದರೊಳಗಡೆ ಏನಿದೆ? ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ ಇದೆ. ಮೈಕ್ರೋಸ್ಪೋರಾಂಜಿಯಂ ಒಳಗಡೆ ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ ಇದೆ. ಅದೇ ತರ ಪೋಲನ್ ಸ್ಯಾಕ್ ಅಲ್ಲಿ ಏನಿದೆ? ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಇದೆ. ಕ್ಲಿಯರ್? Yes. See, this is the dithecous. Easily you can identify. ಎಷ್ಟು ನೀಟಾಗಿ ಕಾಣಿಸ್ತಾ ಇದೆ, the 2 lobes. Dithecous condition. Fine. So, dithecous ಇದೆ ಅಂದ್ರೆ there should be also one more kind. That is called as monothecous. Monothecous. What is the speciality of monothecous here? Monothecous anther, obviously we can understand there is only one lobe. Right? One lobe irutte. One lobe alli of course 2 microsporangium irutte. Correct? That is called as monothecous. Hitira with that one filament is having only one lobe over here. One lobe olgade we can find these 2 microsporangium. Microsporangium. ಸ್ಪೊರಾಂಜಿಯಂ. ಆ ಮೈಕ್ರೋಸ್ಪೊರಾಂಜಿಯಂ ಅಲ್ಲಿ ಏನಿರುತ್ತೆ, ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ ಇರುತ್ತೆ. ಮೈಕ್ರೋಸ್ಪೊರಾಂಜಿಯಂ ಏನಂತ ಕರಿತೀವಿ, ಪೋಲನ್ ಸ್ಯಾಕ್ ಅಂತ ಕರಿತೀವಿ. ಪೋಲನ್ ಸ್ಯಾಕ್ ಒಳಗಡೆ ಏನಿರುತ್ತೆ, ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಇರುತ್ತೆ. ಸೊ ದಿಸ್ is the picture of hibiscus plant. Hibiscus ali you can see this is the lobe. Monothecous condition. One lobe irutte. One lobe alli we can find two microsporangium. So what is the classification of anther? Monothecous and dithecous. Clear? Next. So from that time we learned about anther and its reproductive structures present inside it, correct? Because it is a fertile part, it is having the reproductive structures in it. Fine. Now what is this filament doing? Why filament should be present then? Filament is present for the mechanical support or strength for the anther to hold in a position. Okay, na? So we already know it is a sterile part, long agide slender stalk adu, and proximal end ಅಂದ್ರೆ the below part, proximal end of the filament. See, this is the filament and this is the lobe, correct? This is the lobe. This is attached to the thalamus. Thalamus ಅಂದ್ರೆ this region where the sepals arise, sepals and petals will arise, correct? See, this region, this part which is inverted bell shaped structure idira, this is thalamus athwa receptacle anta kariತೀವಿ. ಈ region ಗೆ attach ಆಗಿರುತ್ತೆ. Filaments will be attached to this part of the flower. All right. See, here you cannot see inside, but it is attached to the thalamus or receptacle part. So that is filament, the long slender like structures. These are called as filaments. Okay? Filaments. Now let us solve few MCQs based on whatever we learned till now. Are you all ready now? Okay, the first question is, male sex organ in a flower is called as. By now I think many of you will recall the answer without the option, but then I'll show you the options. First is stamen, carpel, petal and sepal. Which is the right answer? It is stamen. Correct? Stamen is having anther and filament. That is a male sex organ in the flower. Next question. A broad knob-like fertile part of the stamen is called as, yes, filament, carpel, petal or anther. Which is the right answer? Obviously it should be anther, the male reproductive part. Correct? Next question. Monothecous anther is seen in, example you have to remember. I've said you. Datura, hibiscus, solana or brassica. Which of the plants? It is hibiscus. Correct? So with that, we have completed the structure of a male reproductive organ. Moving on further, that is structure of microsporangium. What is the structure of microsporangium now? What are we focusing on? Andre, see, we learned the male reproductive organ, that is filament and anther, together called as stamen. Filament, anther together called as stamen, correct? ಈ ಆಂಥರ್ ಅಲ್ಲಿ ಸೆಕ್ಷನ್ ಮಾಡಿ ನೋಡಿದಾಗ ನಮ್ಗೆ ಏನ್ ಕಾಣಿಸ್ತು, ಒಂದು ಲೋಬ್ ಅಲ್ಲಿ 4 ಮೈಕ್ರೋಸ್ಪೊರಾಂಜಿಯಂ ಅಥವಾ ಪೋಲನ್ ಸ್ಯಾಕ್ ಕಾಣಿಸ್ತು. Now we are learning this enlarged view of this pollen sac ಅಥವಾ microsporangium. It's also called as what? Pollen sac. All right. Yes. The structure ಅಥವಾ TS of anther ತಗೊಂಡಾಗ we can find the microsporangium is said. Correct. So this young anther, I will just analyze this sentence in the form of a pictorial manner so that you will understand better. ಅರ್ಥ ಆಯ್ತಾ? ಸೊ ಈಗ ಯಂಗ್ ಆಂಥರ್ ತಗೊಂಡಾಗ ಈ ತರ ಆಂಥರ್ ತಗೊಂಡಾಗ ನಾವು ಇನ್ಸೈಡ್ ವಿ ಫೈಂಡ್ ದಿಸ್ ಮೈಕ್ರೋಸ್ಪೊರಾಂಜಿಯಂ ಅಂತ ಹೇಳಿದ್ವಿ, ಕರೆಕ್ಟಾ? ಸೊ ದಿಸ್ ಕನ್ಸಿಸ್ಟ್ ಮೈಕ್ರೋಸ್ಪೊರಾಂಜಿಯಂ ಅಲ್ಲಿ ಏನೇನಿದೆ, ಇಟ್ ಇಸ್ ಹ್ಯಾವಿಂಗ್ ಹೋಮೋಜಿನಿಯಸ್ ಮಾಸ್. ಹೋಮೋಜಿನಿಯಸ್ ಮಾಸ್ ಅಂದ್ರೆ ಇಟ್ ಇಸ್ ಹ್ಯಾವಿಂಗ್ ಆಲ್ ಸಿಮಿಲರ್ ಇನ್ ಸ್ಟ್ರಕ್ಚರ್, ದ ಸೆಲ್ಸ್, ಆಯ್ತಾ? Of meristematic cells. I know meristematic cells are having the capability of dividing continuously. Meristematic cells do speciality nadu. Yen speciality meristematic cells do it can divide continuously throughout its life. It is having maximum dividing capacity. So ee meristematic cells irutte inside this anther. Before the segregation or development of the four microsporangium, there will be some cells like that. Our cells yav tara capability there, it is having this meristematic capability so that it will divide continuously, and further these cells will be called as primary sporogenous cells. Enanta karitive? Primary sporogenous cells. Andre these are having the capability to form spores anta. We call it as microsporangium, correct? The microsporangium agbeku andre primary sporogenous cells should develop first. From where? From the homogeneous mass of cells. Just hafazard age cells irutte inside the anther. These cells mature hegagutte, they will turn into primary sporogenous cells first. Arthaita? Primary sporogenous cells agi, then it will convert into microsporangium. And this sporogenous cells or primary sporogenous cells is being surrounded by the anther wall. Of course, there should be the outermost wall. Idr ಒಳಗಡೆ cells ಇರುತ್ತಲ್ಲ, these cells are homogeneous mass of cells, meristematic cells ಅಂತ ಕೂಡ ಕರಿತೀವಿ, and this will turn into primary sporogenous cells ಆಗಿ turn ಆಗುತ್ತೆ. Okay, so this primary sporogenous cells will further form what? MMC. MMC ಅಂದ್ರೆ ಏನು? Microspore mother cells. Microspore mother cells ಆಗಿ convert. That is diploid in condition. Diploid andre to N. It is having the full chromosomes. Diploid in condition inside the microsporangium andre inside the microsporangium. Inside the microsporangium we will find these primary sporogenous cells converting into MMC. So to summarize, I will tell you again, young anther irutte, it is surrounded by anther wall. Inside we will find homogeneous mass of cells. The homogeneous mass of cells ಏನದು function? It is meristematic in function, andre obviously to divide continuously. So it will divide the cells and form primary sporogenous cells. ಈ primary sporogenous cells ಏನಾಗುತ್ತೆ? It will become MMC. ಏನಾಗುತ್ತೆ? Microspore mother cell ಆಗುತ್ತೆ. ಅದು ಯಾವ ಕಂಡೀಷನ್ ಅಲ್ಲಿ ಇರುತ್ತೆ? ಡಿಪ್ಲಾಯ್ಡ್ ಕಂಡೀಷನ್ ಇರುತ್ತೆ. Did you understand the whole story here? Anther wall is there, inside the small cells are there. These cells are homogeneous mass of cells and they are meristematic in function, and they are called as primary sporogenous cells. These primary sporogenous cells will convert into what? MMC, which is diploid in condition inside these microsporangium. Further ಏನಾಗುತ್ತೆ? How it will produce the male gametes? We will understand in the next slides. See, one second. See, in this picture, you are visualizing these 4 microsporangiums. Again, when I have to zoom it, only one microsporangium, I can see different layers over there. See, these are microspore mother cells. We were talking about these little tiny set of cells till now. See, microspore mother cells. What did we say? Anther wall irutte. Inside the microsporangium, there are these MMC anthers. The MMC form ಆಯ್ತು. Right from the homogeneous mass of cells which were meristematic in function, and they turned into primary sporogenous cells, correct? And these primary sporogenous cells ಏನಾಯ್ತು? Microspore mother cells ಆಯ್ತು, which are diploid in function, which are diploid ಅಂದ್ರೆ 2n ಇರುತ್ತೆ, correct? So these microspore mother cells will next form the male gametes. So surrounding that we can see the middle layers, yellow color ಕಾಣಿಸ್ತಾ ಇದೆಯಲ್ಲ, ಇದೆಲ್ಲ middle layers ಅಂತ ಕರಿತೀವಿ. See, these are middle layers, and there is one more orange circle over there. That is the other layer which is called as the middle layers or the outer middle layers. And there is one green shade over there, dark green, that is endothelium, and the last layer is epidermis. Epidermis, understood? And there is also a tapetal layer over here, a tapetal layer. Fine. So in this image, what and all did we see? There is the TS of young anther, then view of microsporangium, and lastly the third one is the mature dehisced anther andre eno. See, at this stomium anta karitivalla, this in the layer which is more inside alva, concavity ಇದೆ alva, that region is stomium. A stomium region alli enagutte, it will break off, and these MMC iruttalla will turn into pollen grains. How it will turn? Again, it is having some process we will learn. So it will break and it will be moving out from the anther. Did you understand? So this is the diagrammatic representation of the young anther and the enlarged view of microsporangium and the dehisced anther. And the breakout at the side, stomium regionally, it will break and all the pollen grains will be released out. Okay. Next, we will learn about the anther wall layers. So in the previous image, we saw the different layers, correct? So anther wall consists of mainly 4 layers, out of which epidermis is said to be the outermost layer. Okay, it is the outermost layer, and it is one-celled thick layer. Andre, it is having one cell and it is also thick layer. It is one-celled thick layer. The thin-walled cells present between 2 pollen sacs. Outermost thick layer. And there are 2 pollen sacs in between one lobe, correct? Between there are thin cells present. Yake? Because to easily break out, to easily get dehisced. Adike, it is having a thin wall over there. Wait, I'll show you. See here. Nodi, E region alli, it will have a thick layer. Okay, it is having a thick epidermal layer. But when it is coming in between the 2 pollen sacs or 2 microsporangium, it is having a thin layer. Reason? Because it is able to break this layer and allow all the pollen grains out. Break at this region, it should tear or it should get dehisced. Adike, it is having a thin layer. That thin layer is called as stomium. What is that thin layer called as? It is called as stomium. That is what it is said. Thin walled cells are present between 2 pollen sacs. It is called as stomium. Now, what is this function? As we said, it is nothing but helping in pollen sacs dehiscence, to move the pollen sacs out of the anther wall layer. See, this is the stomium. Got it? This is the outermost epidermal layer. It is thick. At the center or between two pollen sites, it is becoming thin. The wall layer is thin. So that region is called as stomium. Stomium function is just to dehisce the pollen grains. Got it? This is the stomium. Next, the inner wall layer. Inner wall layer yaudu? See, this is the epidermis, correct? Epidermis ಒಳಗಡೆ layer na endothecium ಅಂತ ಕರಿತೀವಿ. Now we are learning about this layer. Endothecium ಬಗ್ಗೆ ಓದ್ತಾ ಇರೋದು ಇವಾಗ ನಾವು. What is this endothecium? Of course, it is inner to the epidermis. Inner to the epidermis. And these cells have the cellulosic fibrous thickenings. These cells, they are having cellulose content in it and they are fibrous in nature. Okay, these cells are cellulosic fibrous in nature. Okay, cellulosic fibrous in nature, which arise from inner tangential walls. Okay, which arise from inner tangential walls. Inner tangential walls of what? Of anther. So it has been arise from the inner tangential walls of anther, which helps in dehiscence of anther. Again, along with the stomium region, even endothecial layer is helping in dehiscence of anther due to their hygroscopic nature. Hygro andre, so water-loving nature, hygroscopic nature. Okay, whenever there is this encountering of water droplets or due to humidity or moisture, it will get dehisced easily. Alright, so this is the function of endothecium layer, which is inner to the epithelium or epidermal layer. Fine. See, this is the epidermis, the outermost region, and inner to that we can find this endothecium layer. Right? So even this is helping in dehiscence of anther. Got it? Next, the third layer is middle layer. Which is the middle layer now? See, first one is epidermis. Then comes the endothecium. Then we can see these orange band-like structures, no? These are middle layers. Okay, these are middle layers. And inner to it is tapetum. Inner to that is microspore mother cells. Got it? So, middle layers are usually 1 to 3 layers of cells. These layers degenerate at maturity. At maturity, what happens when they are in the stage of releasing the pollen grains into the outside environment? Again, middle layers will indirectly get degenerated. They will start to degenerate and will help in easy dehiscence of the pollen grains with the help of the stomium and the endothecial layers. Endothecial layers. Okay, so this is the function of middle layers. Middle layers and the HT there is about 1 to 3 layers. You'll not put them. It is very thick because it is arranged in 1 to 3 layers. Right? So this is the third one. Which was the first one? Epidermis. Second is endothecium. Third one is middle layers. And this is the fourth layer. That is tapetum. Okay, fourth layer is tapetum. What is the function of tapetum now? Tapetum involves a nourishment of these developing pollen. Still now, ಈ ಎಪಿಡರ್ಮಿಸ್ ಎಂಡೋಥೀಶಿಯಂ ಮಿಡಲ್ ಲೇಯರ್ಸ್ ಈ ಎಲ್ಲಾನು ನಾವೇನ್ ಅರ್ಥ ಮಾಡ್ಕೊಂಡ್ವಿ, these three are present only for helping the dehiscence of pollen grains ಅಷ್ಟೇ, ಕರೆಕ್ಟಾ? But this tapetum plays a very important role. ಈಗ pollen grains dehisce ಆಗಬೇಕು, okay fine, it should move outside the environment. But pollen grains develop ಆಗಬೇಕಲ್ವಾ, it should get some nourishment. Who is that person giving nourishment to the pollen grains? That is tapetum. Okay, so tapetum is the innermost layer. This is the innermost layer of the anther wall which surrounds the sporogenous tissue. Sporogenous tissue andre primary sporogenous tissue which we talked about, which will turn into MMC, microspore mother cell, correct? So other sutta iro layer tapetum layer. Yaake adu suttane ide, yaake adu next endothelium ಆದ್ಮೇಲೆ ಇಲ್ಲ, middle layers ಆದ್ಮೇಲೆ ಇಲ್ಲ, because as it is next to sporogenous tissue, easy nourishment ಹೋಗಬಹುದು, correct? So this tapetal layer will give nourishment to the developing pollen grains. Pollen grains ge easy nourishment sigate. That is why the structure is also been so beautifully arranged by the creation. So the creation is wonderful, correct alva? So that layer itself is present next to it because it is having some prominent function over there. Okay, every layer or every structure which is present has one or the other reason which is very useful for the developmental process, correct? So your tapetal layer will help in the nourishment of the developing pollen grains. So iga, what is the speciality of tapetum? How are these cells nourishment kodbeku andre it should have some storage in it, correct? It should have some storage in it so that it can provide to other tissue over there. So what is it having? It is having a dense cytoplasm. Cytoplasm dense, thick agide, so that it is creating a room or creating a space for more and more nourishment in it, correct? And it is having more than one nucleus. Come on, in cell we have only not one nucleus, one cell only one nucleus. Now it is that this tapetal cells, we are seeing that it is having more than one nucleus. How is that possible? Ek sadhyada, because during this mitosis cell division there is this stage called as cytokinesis, correct? Cytokinesis ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಈ ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವೈಡ್ ಆಗಿರುತ್ತಲ್ಲ ಕ್ಯಾರಿಯೋಕೈನೆಸಿಸ್ ಪ್ರಾಸೆಸ್ ಅಲ್ಲಿ, ಕ್ಯಾರಿಯೋಕೈನೆಸಿಸ್ is nothing but splitting of nucleus into one or two, more than two, correct? So ಈ ಕ್ಯಾರಿಯೋಕೈನೆಸಿಸ್ ಆದ್ಮೇಲೆ followed by that there should be the stage called as cytokinesis. ಈ cytokinesis ಆಗಿಲ್ಲ ಅಂದಾಗ ಈ ತರ ಮಲ್ಟಿ ನ್ಯೂಕ್ಲಿಯೇಟ್ ಸ್ಟೇಜ್ ಆಗುತ್ತೆ. ಸೊ ಈ tapetal cells ಅಲ್ಲೂ ಇದೇ ತರ ಆಗಿದೆ. ಇವಾಗ ಏನಾಗಿದೆ, ಇಲ್ಲಿ ಸೆಲ್ ಡಿವಿಷನ್ ಟೈಮ್ ಅಲ್ಲಿ ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವೈಡ್ ಆಗಿದೆ, ಬಟ್ ಇಟ್ ಡಸ್ ನಾಟ್ ಹ್ಯಾವ್ ಅ ಸೆಪರೇಟ್ cell walls for it. It is being dumped into one cell. Okay, it has not created the boundaries. It is present all together at one cell. Adike it is having more than one nucleus with the dense cytoplasm. Again, more than one nucleus and its efficiency increases. One nucleus cells it is having a beautiful efficiency. Iga more than one nucleus andre it is having more capacity to function. See here, we can find this tapetal layer, right? So which is the outermost layer? Epidermis. Next to it, endothelium. Next to it, middle layer. And then tapetal layers. And inner to it is MMC. So 4 anther wall layers. You all must know by now. Yavyadu? First one is epidermis. See? Different color, wait. See, epidermis. First one. Next, endothelium. Second layer, innermost layer. And inner to endothelium, we can find middle layers. And inner to middle layer, we can find MMC. That is microspore mother cells. Before that, we have this tapetal layer. Okay. So this will become 5 and this is 4th one. Again, from first outermost epidermis, inner to epidermis is endothelium. Inner to endothelium is middle layer. Inner to middle layer is tapetal layer. Tapetal layer creates a surrounding boundary to these MMC microspore mother cells. These are the 4 anther wall layers which you have to remember. More questions will be asked based on this, and this chapter itself is having more weightage, as I said in the previous session, right? So please concentrate more on this chapter with respect to board exam or also your CET as well as NET exam, okay? So NEET exam, and this is Stomium. Stomium and Reno. There is this inner groove-like structure. We have learned this structure, groove-like structure called stomium, which is having purely functioning in dehiscence. Correct? So these are the 4 anther-volutes. And of course, inner to that, we have this tissue, sporogenous tissue. That MMC and that is only sporogenous tissue. And these sporogenous tissue, primary sporogenous tissue, will be giving rise to MMC. This MMC, microspore mother cell, will convert into microspore tetrad. Tetrad andre tetra, 4 in number. Microspore mother cell will give into 4 cells, microspore tetrad stage. So each one, microspore tetrad, each one will have the potential to develop into PMC. Sorry, MMC will be converted into PMC. Andre microspore mother cell will give rise to microspore tetrad. This microspore tetrad, each one has that potential to give rise into pollen mother cell, PMC. Pollen mother cell andre no. Pollen mother cell is the cell which gives rise to pollen grains. Correct? So this whole process is called as microsporogenesis. And then microsporogenesis, micro, small, spores andre small tiny structures. The genesis andre generating. So this process is nothing but generating microspores. That only when we have to put it into a definition, how will we define it? The process of formation of microspores. Process of formation called microspores. Form ಆಗ್ತಿರೋದು, generate ಆಗ್ತಿರೋದು. From what? From PMC, pollen mother cell. Or from MMC, microspore mother cell through the cell division called as meiosis is called microsporogenesis. Very important definition. Remember this. So what is microsporogenesis? It is the process of formation of microspores from a PMC athwa NMC before that through the cell division meiosis. Meiosis is called microsporogenesis. Got it? So this is the formation. This is in the track of the formation of pollen grains, that is male gametes. We are learning now. We are touching into that topic now. Alright. So these microspores formed from the microsporogenesis are arranged, again we are coming back to this point, are arranged into microspore tetrad. 4 cluster ಆಗಿರುತ್ತೆ, group ಆಗಿರುತ್ತೆ, ಅದನ್ನ microspore tetrad ಅಂತ ಕರಿತೀನಿ. ಈಗ PMC, what we talked about PMC here, meiosis 1 stage ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, diploid 2n condition ನಲ್ಲಿ 2 cell ಆಗಿ divide ಆಗುತ್ತೆ. Further tetrad stage ಗೆ ಬರಬೇಕು ಅಂದ್ರೆ meiosis 2 ಆಗುತ್ತೆ. ಅಲ್ಲಿ meiosis 2 ಏನಾಗುತ್ತೆ, ಈ diploid condition it will turn into a haploid condition and convert into 4 cells with each having n nucleus. And these nucleus, which is combined in the form of 4 tetrad stage it will segregate or separate into 4 different pollen grains. This is how exactly the pollen grains are formed inside the anther, and that too inside the sporogenous tissue, the innermost layer of the anther, and that is why we call it as the reproductive structure. The anther is called as reproductive structure. Because it is producing the pollen grains. What are these pollen grains? Pollen grains are nothing but the male gametes. Correct? So this is how the whole story of how pollen grains develop inside the anther. So when we learn, it is like a huge one. But this happens, in a fraction of seconds inside the plant. Unknowingly. But when we have to learn, we will learn it as a sequential manner. One step by step flow chart right? But this whole process is happening in plants. This is how the small and the inside that this porogenous tissue is able to divide into pollen grains. How wonderful is it? Correct. So this is the process of formation of pollen grains. See, first diploid condition irutte meiosis agi microsporogenesis alli meiosis ಆಗ್ತಾ ಇದೆ ಅದಕ್ಕೆ ಡಿಪ್ಲಾಯ್ಡ್ ಕಂಡೀಷನ್ ಆಗುತ್ತೆ ಆಮೇಲೆ meiosis 2 ಆದಾಗ ಏನಾಗುತ್ತೆ ಈ ಡಿಪ್ಲಾಯ್ಡ್ 2n ಇರೋದು nn ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗಿ 4 tetrad conditional ಆಗುತ್ತೆ again then it will separate into 4 different pollen grains, right? So with that we will learn as to what are these pollen grains, how is the structure of this pollen grain, okay, what are the uses or advantages or disadvantages of these pollen grains anta. Okay, so these pollen grains hegide nodakke usually they are spherical in shape. Yav shape? Spherical, round in shape. They are measuring in micrometers. Micrometers is to 25 to 50 micrometers, not even visible. Even in microscopic view, it is very tiny. Okay, after utilizing all the process in the section. So it is 25 to 50 micrometer in diameter. Understood? And the cell wall of pollen grain. Pollen grain spherical shape anta helidvi. It is having that cell wall, outermost cell wall, that is called a sporoderm. Okay, the cell wall of pollen grain enanta karitide sporoderm. What is the cell wall of pollen grain called as? Sporoderm. Remember the terms. Terms are very important. Cell wall of pollen grain is called as sporoderm, which consists again of 2 layers. The cell wall is having 2 layers again. 2 layers ಏನಂತ ಕರಿತೀವಿ ಅಂತ we will learn. The hard outer layer is called as exine. Okay, this is a hard layer, outer layer of the pollen grain. These are two together called as sporoderm. The sporoderm is having 2 layers. What are those 2 layers? Outermost layer is exine, hard layer. Inner layer is called as intine. Inner layer is called as what? Intine anta karitivi. Remember, sporoderm has 2 layers, outermost hard layer, exine, and innermost is intine. Okay? Then, see, you can visualize in this picture. Hard and ಎಷ್ಟು ಕ್ರೂಕಿ in nature. Image ನೋಡಿ ಎಷ್ಟಿದೆ ಹೆಂಗಿದೆ ಅದು ತುಂಬಾ rough surface ಇದೆ. So this hard outer layer exine is like this in visualization. Okay? And intine is the inner soft layer when compared to exine. Okay? This is exine and intine. Exine, when we have to learn about exine now. Exine is made up of sporopollen. The hard layer anta ಹೇಳಿದ್ವಿ ಅದು hard ಇದೆ exine ಅಂತ. ಯಾಕೆ hard ಇದೆ? Because it is having sporopollen in. Why exine is hard? Because it is having this substance called or chemical called as sporopollenin. Important term. Sporopollenin. ಈ ಸಬ್ಸ್ಟೆನ್ಸ್ ಏನ್ ಮಾಡುತ್ತೆ? It is resistant to organic materials. It is resistant. Why it has to be resistant to organic materials here? See, this is the sectioning of the pollen grain. This exine nodi ishtu rough surface ide. Intine is a smooth inner surface. And inside that we have nuclei. You can see there are 2 cells. One is bigger cell, one is smaller cell. We will understand that. Bigger one and a smaller cell with 2 nuclei there. And there is also thickening of cellulose inside it. Fine. So before that, we learned that exine is made up of sporopollenin and this is a fatty substance. and which is most resistant organic material. Resistant organic material. From what it is resistant? It is resistant to high temperatures or strong acid athwa alkali. And till now, there was no enzyme being discovered to degrade this enzyme layer. ಅಂದ್ರೆ ಈ ಎಕ್ಸೈಮ್ ಲೇಯರ್ ನ ಡಿಗ್ರೇಡ್ ಆರ್ ಟು ಡಿಜೆನರೇಟ್ ಮಾಡುವಂತ ಎನ್ಸೈಮ್ ಇನ್ನು ಡಿಸ್ಕವರಿ ಆಗಿಲ್ಲ ಅಥವಾ ಇನ್ನು ಆ ಎನ್ಸೈಮ್ ಇಲ್ಲ, ಅಂತಹ ಎನ್ಸೈಮೇ ಇಲ್ಲ. ಬಿಕಾಸ್ ಇಟ್ ಇಸ್ ಸೋ ಮಚ್ ರೆಸಿಸ್ಟೆಂಟ್ ಟು ಆಲ್ ದಿ ಆರ್ಗ್ಯಾನಿಕ್ ಮಟೀರಿಯಲ್ಸ್ ಆರ್ ಟು ಆಲ್ ದ ಅದರ್ ಹಿಂಡ್ರೆನ್ಸ್ ಲೈಕ್ ಹೈ ಟೆಂಪರೇಚರ್ ಆಗಿರ್ಬೋದು, ಸ್ಟ್ರಾಂಗ್ ಆಸಿಡ್ಸ್ ಆಗಿರ್ಬೋದು ಅಥವಾ ಆಲ್ಕಲಿ ಆಗಿರ್ಬೋದು, ಇದು ಯಾವ್ದ್ರಿಂದನೂ ಎಕ್ಸೈಮ್ ನ ಡ್ಯಾಮೇಜ್ ಮಾಡಕ್ಕೆ ಆಗಲ್ಲ. So that hard or that capacity the enzyme layer has. Got it? Sporopollen is an important term. So because of the sporopollen, pollen grains of the past plants are well preserved as fossils ಆಗಿ ಪ್ರಿಸರ್ವ್ ಮಾಡಬಹುದು. ಇದನ್ನ ಏನ್ ಮಾಡಬಹುದು ನಾವು, ಇದು ಇಷ್ಟು ಹಾರ್ಡ್ ಇರೋದ್ರಿಂದ, because of this enzyme which is very hard and easily degenerate ಆಗಲ್ಲ ಅಥವಾ ಡಿಸ್ಟ್ರಾಯ್ ಆಗಲ್ಲ ಅಥವಾ ಡ್ಯಾಮೇಜ್ ಆಗಲ್ಲ, ಇದನ್ನ ನಾವೇನ್ ಮಾಡಬಹುದು, ಫಾಸಿಲ್ ರೂಪದಲ್ಲಿ ಇದನ್ನ ಪ್ರಿಸರ್ವೇಷನ್ ಮಾಡಬಹುದು. we can preserve it. One of the important plants ಅಂತ ಇರುತ್ತೆ which are threatened ಅಥವಾ ಎಕ್ಸ್ಟಿಂಕ್ಟ್ ಆಗುವ ಚಾನ್ಸಸ್ ಇರುತ್ತೆ. ಅಂತಹ ಪ್ಲಾಂಟ್ಸ್ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ನ ನಾವೇನಾದ್ರೂ ಪ್ರಿಸರ್ವ್ ಮಾಡಿದ್ರೆ we can still cultivate these plants. Even the conditions are not favorable in the outside environment, we can bring those pollen grains into the laboratory conditions and we can start to cultivate it. Or tissue culture ಮಾಡಬಹುದು, ಏನ್ ಬೇಕಾದ್ರೂ ಮಾಡಬಹುದು ಇದೆಲ್ಲ. So because of this enzyme, it is having so much uses. Correct? Thanks to enzyme, so that we are having the chance to preserve it and also do more research on it. Correct? Or also we can preserve so many plants for that reason as fossils. Correct? So pollen grain enzyme, we can also find the germ pores in it. Enzyme alli small pores kuda kaaniಸುತ್ತೆ. I will show you in this image. See, 1, 2, 3. Few number of germ pores we can visualize in the enzyme layer. ಅಲ್ಲಿ ಇಲ್ಲಿ germ pores ಅಂತ ಇರುತ್ತೆ. ಈ germ pores ಏನಕ್ಕೆ ಬೇಕು? What are these germ pores? Germ pores where sporopollen is absent. Wow. ಈಗ ಸ್ಪೋರೋಪೊಲಿನ್ ಇಷ್ಟು ಇಂಪಾರ್ಟೆನ್ಸ್ ಕಲ್ತ್ಕೊಂಡು ನಾವು ಇಲ್ಲಿ ಸ್ಪೋರೋಪೊಲಿನೇ ಇಲ್ಲ ಈ ಪಾರ್ಟ್ ಅಲ್ಲಿ, ಒಂದೊಂದು ಜಾಗದಲ್ಲಿ ಎಕ್ಸೈಮ್ ಅಲ್ಲಿ. ಯಾಕಿಲ್ಲ? ವೈ? We will learn it now. See, this is the germ pore. This is the germ pore and this is the hard enzyme layer. ಈ ತರ ಡಿಫ್ರೆಂಟ್ ಆಗಿ ಸ್ಟ್ರಕ್ಚರೈಸ್ಡ್ ಆಗಿರುತ್ತೆ, ಎಲ್ಲ ಒಂದೇ ತರ ಇರಲ್ಲ ಎಕ್ಸೈಮ್ ಲೇಯರ್ಸ್. ದೇರ್ ಆರ್ ಸೋ ಮೆನಿ ಡಿಫ್ರೆಂಟ್ varying in different species. Okay. The intine, what happens to intine now? Intine is the innermost layer of the sporoderm of pollen grain. This is made up of cellulose and pectin. What is it made up of? Cellulose and pectin. Intine is made up of cellulose and pectin. These are the two pigments, or these are the two important substances which are present inside the intine. That is cellulose and pectin. Okay, with that we have covered the structure of the pollen grain and what are the important substance or organic materials present in the layers of the pollen grain. Okay, so now we will continue with the development of male gametophyte. How does the male gametophyte develops? So we already know this story now. What is the story? Microspore is the first cell of the male gametophyte. We know that. Microspore tetrad agate. Allinda PMC inda athwa MMC inda pollen mother cell athwa MMC microspore mother cell inda ಮೈಕ್ರೋಸ್ಪೋರ್ ಟೆಟ್ರಡ್ ಆಗುತ್ತೆ. ಆ ಮೈಕ್ರೋಸ್ಪೋರ್ ಟೆಟ್ರಡ್ ಈಚ್ ಮೈಕ್ರೋಸ್ಪೋರ್ ಆಗುತ್ತೆ. ಫೈನ್. ಈ ಮೈಕ್ರೋಸ್ಪೋರ್ನೇ ನಾವೇನಂತ ಕರಿತೀವಿ, ಫಸ್ಟ್ ಸೆಲ್ ಆಫ್ ಮೇಲ್ ಗೆಮೆಟೋಫೈಟ್. ಫಸ್ಟ್ ಸೆಲ್ ಆಫ್ ದಿ ಎಂಬ್ರಿಯೋ ಯಾವುದು? ಝೈಗೋಟ್. ಕರೆಕ್ಟಾ? ಅದೇ ತರ ಫಸ್ಟ್ ಸೆಲ್ ಆಫ್ ದಿ ಮೇಲ್ ಗೆಮೆಟೋಫೈಟ್ ಇಸ್ microspore. Okay, so the germination of microspore starts inside the anther itself. Germination of microspore starts inside, andre in situ, in the mother place, andre inside the anther wall itself, andre inside the tapetal layer, sporogenous tissue. So this microspore will be best defined as partially developed male gametophyte. Andre it is partially developed male gametophyte anta kuda karibodu. Microspore can be also called as partially developed male gametophyte. What is it called as? Partially developed male gametophyte. Partially, inna fully developed, fully mature agilla. Fully mature yavaga agutte we will get to know now. So eega microspore germinate aguttalla inside the sporogenous tissue or inside the tapetal layer, then it is called as partially athwa halfly developed male gametophyte anta kariತೀವಿ. Then these microspores will then divide into large vegetative cell, small generative cell. As I said, ಅಲ್ಲಿ ಒಂದು last slides ಅಲ್ಲಿ image ನೋಡಿದ್ವಿ, ಅದ್ರಲ್ಲಿ ನಾನು ಹೇಳಿದೆ this was the pollen grain. ಅದರಲ್ಲಿ ಈ ತರ ಸೆಪರೇಷನ್ ಆಗಿತ್ತು, ಒಂದು ದೊಡ್ಡ ಸೆಲ್ ಒಂದು ಚಿಕ್ಕ ಸೆಲ್ ಹಾಗೆ. ಈ ಚಿಕ್ಕ ಹ್ಯಾಡ್ ಒಂದು ನ್ಯೂಕ್ಲಿಯಸ್ ಇದೆ. ಸೊ ದೀಸ್ ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ ಏನಾಗುತ್ತೆ, ವಿಲ್ ಡಿವೈಡ್ ಮೈಟೋಟಿಕಲಿ. ಮೈಟೋಸಿಸ್ ಅಂದ್ರೆ ಏನು? ಮೈಟೋಸಿಸ್ ಅಂದ್ರೆ ಆಬ್ವಿಯಸ್ಲಿ ಇಟ್ ಇಸ್ ಎ ರಿಡಕ್ಷನಲ್ ಡಿವಿಷನ್. ಕರೆಕ್ಟಾ? ಸೊ ಈ ರಿಡಕ್ಷನಲ್ ಡಿವಿಷನ್ ಆದಾಗ ಏನಾಗುತ್ತೆ, ಈಕ್ವಲ್ ಆಗಿ ಡಿವಿಷನ್ ಆಗ್ತಾ ಇಲ್ಲ. ಇಲ್ಲಿ ಈಕ್ವಲ್ ಆಗಿ ಡಿವಿಷನ್ ಆಗ್ತಾ ಇಲ್ಲ, ಬಟ್ ಇಟ್ ಇಸ್ ಡಿವೈಡೆಡ್ ಇಂಟು ಅ ಲಾರ್ಜರ್ ಸೆಲ್ and a small cell building. Now what is this large cell and what is this small cell called as? Large cell na navu vegetative cell anta karitivi. What do we call it as? Vegetative cell. Okay, and small cell is called as generative cell. Small cell is called as generative cell. Vegetative cell and generative cell. Got it? Did you understand till here? So, ಆಮೇಲೆ ಏನಾಗುತ್ತೆ, the generative cell. See here, you can see there is very small generative cell and the large vegetative cell. Correct? ಈ generative cell ಅಲ್ಲಿ ಏನಿರುತ್ತೆ, there is spindle shape. ಯಾವ shape ಇದೆ? ಇದು spindle shape ಇದೆ. Generative cell is not circular in shape, but it is a spindle shape. See? Spindle shape or kite shape ಇದೆ. With ಒಳಗಡೆ ಏನಿರುತ್ತೆ, ಡೆನ್ಸ್ ಸೈಟೋಪ್ಲಾಸಮ್ ಇರುತ್ತೆ. With dense cytoplasm and a centrally located nucleus. See, generative cell is very small and this is in spindle shape with dense cytoplasm and nucleus. Got it? So, idralli enide, it is also having cytoplasm, vegetative cell alli. Along with cytoplasm, it is having nucleus. That nucleus is called as tube nucleus. Yake tube nucleus anta karitivi, we will get it now. So here, idenagtide, now we are learning the internal structure of the pollen grain, the exino, intino. ಒಳಗಡೆ ಹೇಗೆ ಡಿವೈಡ್ ಆಗ್ತಿದೆ ಮೈಟೋಟಿಕಲಿ, we are learning now. Okay. So see again what happens. Generative cell and vegetative cell together irutte. ಅದಾದ್ಮೇಲೆ ಏನಾಗ್ತಾ ಇದೆ, here further you can see this tube-like structure generating here. The tube-like structure has, see, male cells and the tube nucleus. Tube nucleus from the generative cell will be here. Male cells elinda bantu? Male cells has come from generative cell. Generative cell inda male nucleus ಏನಾಗಿದೆ, ಮೇಲ್ ಸೆಲ್ ಆಗಿ ಡೆವಲಪ್ ಆಗಿದೆ. 2 ಮೇಲ್ ಸೆಲ್ಸ್ ಅಥವಾ 2 ಸ್ಪರ್ಮ್ ಸೆಲ್ಸ್ ಆಗಿ ಡೆವಲಪ್ ಆಗುತ್ತೆ. ದಿಸ್ ಇಸ್ ದ ಪೋಲನ್ ಗ್ರೇನ್. ಪೋಲನ್ ಗ್ರೇನ್ ಇಂದ ಪೋಲನ್ ಟ್ಯೂಬ್ ಆಗುತ್ತೆ. ಯಾವಾಗ ಪೋಲನ್ ಟ್ಯೂಬ್ ಆಗ್ಬೇಕದು? ಆಫ್ಟರ್ ದ ಪಾಲಿನೇಷನ್. ಅಂದ್ರೆ ಏನು, Transfer of pollen grain from anther of the male gamete to the stigma of the female gametophyte. Correct? So when this anther pollen grains are transferring from the anther to the stigma, ಈ pollen grains ಹೋಗಿ this pollen grain halfly mature ಆಗಿರುತ್ತಲ್ಲ, ಇದು ಹೋಗಿ whenever it hasn't fallen on the stigma, ಏನಾಗುತ್ತೆ stigma ಮೇಲೆ ಬಿದ್ದ ತಕ್ಷಣ there will be that induction inside the pollen grain to develop this pollen tube. So ಈ pollen tube development ಇಂದಾನೆ it will send the sperm cells ಅಂದ್ರೆ male cells, male nucleus, it will send the nucleus inside the female reproductive organ. Correct? This is why this pollen tube is being generated. Gotaita? So here you have to know difference between generative cell and vegetative cell. Vegetative andre other their own cell. Generative cells andre something which is having the male cells, male gametes there. So this pollination ಯಾವಾಗ ಆಗುತ್ತೆ? either two celled stage ಇದ್ದಾಗ ಆಗುತ್ತೆ. ಅಂದ್ರೆ two celled ಅಂದ್ರೆ ಈ ತರ ಇರೋ ಸ್ಟೇಜ್ ಅಲ್ಲಿ pollination ಆಗುತ್ತೆ. ಆ ಟೈಮ್ ಅಲ್ಲಿ these pollen grains will move from anther to stigma, ಹೋಗಿ ಅಟ್ಯಾಚ್ ಆಗುತ್ತೆ. ಆ ಟೈಮ್ ಅಲ್ಲಿ it can go, then it will be a successful pollination. two cell stage ಅಂದ್ರೆ with the vegetative cell ಅಥವಾ generative cell ಇವೆರಡು ಇರುತ್ತಲ್ಲ ಆ ಸ್ಟೇಜ್ ಅಲ್ಲಿ ಹೋಗ್ಬೋದು. And that is happening in most of the 60% flowers. 60% angiospermic plants ಅಲ್ಲಿ ಇದೇ ತರನೇ ಆಗೋದು. two celled condition ಅಲ್ಲೇನೆ it will go fall on the stigma. That is generative cell, vegetative cell. Remaining ಇದೆಲ್ಲ 60 ಅಂದ್ರೆ 40% remaining plants ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ? 3 celled stage ಅಲ್ಲಿ it will go pollinate. 3 cell stage ಅಂದ್ರೆ ಏನಾಯ್ತು ಈಗ? Come on, we have learned only about generative and vegetative cell. ಇನ್ನೊಂದು cell ಯಾವುದು? Don't worry. See, vegetative cell in common ಅದರದೇ cell. ಈ generative cell ಏನಾಗುತ್ತೆ? This generative cell will develop into male cells. Generative cell will develop into male cell. 2 male cell ಆಗಿ develop ಆಗಿರುತ್ತೆ. ಅದಕ್ಕೆ ಅದನ್ನ 3 cell stage ಅಂತ ಕರಿತೀವಿ. ಆಲ್ರೆಡಿ ಅದು ಡೆವಲಪ್ ಆಗ್ಬಿಟ್ಟಿರುತ್ತೆ. ಬಟ್ 2 cell stage ಅಲ್ಲಿ ಅದು generative cell ಆಗೇ ಇರುತ್ತೆ. pollen tube ಆದ್ಮೇಲೆ ಮೇಲ್ 2 sperm cell send ಮಾಡುತ್ತೆ. ಬಟ್ 3 cell stage ಆದ್ರೆ ಏನಾಗುತ್ತೆ? ಮುಂಚೆ before it is pollinating only, it will be developed into 2 male cells ಅಥವಾ 2 male gametes ಆಗಿ ಡೆವಲಪ್ ಆಗಿರುತ್ತೆ. ಸೊ ಅದು ಆಗೋದು 40% ಅಷ್ಟು ಚಾನ್ಸ್ ಇದೆ. ಮ್ಯಾಕ್ಸಿಮಮ್ 2 cell stage ಅಲ್ಲೇ ಪಾಲಿನೇಷನ್ ಆಗುತ್ತೆ. ಗಾಟ್ ಇಟ್? ಸೊ ಫರ್ದರ್ ಡೆವಲಪ್ಮೆಂಟ್ ಹೇಗೆ ಪಾಲಿನೇಷನ್ ಅಥವಾ ಮೇಲ್ ಅಂಡ್ ಫಿಮೇಲ್ ಗೆಮೆಟ್ ಹೆಂಗ್ ಫ್ಯೂಸ್ ಆಗುತ್ತೆ, ದಟ್ ಈಸ್ ಹ್ಯಾಪನಿಂಗ್ ಇನ್ ದ ಸ್ಟಿಗ್ಮಾ ಅಂದ್ರೆ ಫಿಮೇಲ್ ಗೆಮೆಟೋಫೈಟ್ ಅಲ್ಲೇ ಆಗುತ್ತೆ. ನೆಕ್ಸ್ಟ್ ಫರ್ದರ್ ಡೆವಲಪ್ಮೆಂಟ್ ಆಫ್ ದ ರಿಪ್ರೊಡಕ್ಷನ್. ಸೊ ದಿಸ್ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಎಕ್ಸ್ಪ್ಯಾಂಡ್ಸ್ ಬೈ ಈ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಹೋಗಿ ಅಟ್ಯಾಚ್ ಆಗುತ್ತಲ್ಲ ಸ್ಟಿಗ್ಮಾ ಮೇಲೆ, ಅವಾಗ ಏನಾಗುತ್ತೆ? ಇಟ್ ವಿಲ್ ಎಕ್ಸ್ಪ್ಯಾಂಡ್, ಇಟ್ ವಿಲ್ ಸ್ಟಾರ್ಟ್ ಟು ಎಕ್ಸ್ಪ್ಯಾಂಡ್ ಇಟ್ಸ್ ಡಯಾಮೀಟರ್ by absorbing the moist surface liquid. Some moist irutte stigma mele, it will absorb that and it will start to bulge. A stigma yen provide madutte so that the pollen grain starts to bulge? It will have boron, sugar matte amino acids anta chemical substance irutte stigma mele. So whenever this pollen grain goes sit on the stigma, what it will do? It will absorb all these chemical substances. That is why it will get expanding. So that is what systematically shown together in ತೋರಿಸಿರೋದು. Microsporocyte ಅಂದ್ರೆ again this microspore, this one microspore the cells in the meiosis ಆಗುತ್ತೆ. ಅಲ್ಲಿ pollen sac, microsporangium in the dehiscence ಆಗಿ pollen sac in the pollen grains will come. Each microspore tetrad in the microspore ಆಗುತ್ತೆ. With the help of mitosis, then the pollen grain will develop into 2 celled stage, that is generative cell ಮತ್ತೆ vegetative cell ಆಗಿ develop ಆಗುತ್ತೆ. Generative cell and vegetative cell, and this will go sit on the stigma, the female gametophyte. ಅವಾಗ ಏನಾಗುತ್ತೆ? sperm cells along with the nucleus, tube nucleus will generate by absorbing all these liquid on the stigma. and it will start to release the nucleus, male cells or the sperms into the female gametophyte, that is stigma style in the ovary gobekala, that in the tube format, pollen tube ಒಳಗಡೆ ಇಂದಾನೆ it will go reach till the ovary inside the ovule. So pollen tube illinda start agi illi tanka beliyuತ್ತದೆ pollen tube. So that is how the sperms have been transferred from the stigma to the ovule, to ovule of the ovary. This is how the process is happening. Got it? So this intine ಏನಾಗುತ್ತೆ ಇವಾಗ in germ pore, we learned germ pore, correct? ಆ germ pore region ನಲ್ಲೇನೆ pollen tube generate ಆಗುತ್ತೆ, because we learned that sporopollen is a very hard substance, ಅದನ್ನ ಏನು degrade ಮಾಡಕ್ ಆಗಲ್ಲ ಅಂತ. ಅದಕ್ಕೆ it has created some space here and there. 2-3 germ pores ಇರುತ್ತೆ. ಆ germ pores ಇಂದಾನೆ pollen tube germinate ಆಗೋದು. Got it? So this intine will come out in the form of pollen tube from germ pores. So intine ಒಳಗಡೆ layer ಇರುತ್ತಲ್ಲ, intine, from that the pollen tube germinate from the germ pores. So the pollen grains are either monocyphinous ಅಥವಾ polycyphinous. Pollen grains monocyphinous ಅಥವಾ polycyphinous ಅಂದ್ರೆ ಏನು? Pollen grains ಒಂದು pollen tube generate ಮಾಡಬಹುದು with one germ pore, ಅಥವಾ it can also generate more than one pollen tube from 2 or 3 germ pores. So that is polycyphinous. Monocyphinous, polycyphinous. ಈಗ pollen grain ಗೆ germ pores ಇದೆ. One of the germ pore in the pollen tube ಬಂದ್ರೆ monocyphinous. ಎಲ್ಲ germ pores 2 or 3 germ pores in the pollen tube generate ಆದ್ರೆ then it is called as polycyphinous. Mono, poly, easy. Okay. So monocyphinous ಯಾವ plants ಅಲ್ಲಿ ನಾವು ನೋಡಬಹುದು? Cucurbitaceae family ಅಲ್ಲಿ ನೋಡಬಹುದು. What are the plants coming under Cucurbitaceae? Like cucumber, watermelon, ಇವೆಲ್ಲ Cucurbitaceae family ಬರುತ್ತೆ. ಈ ಕಂಡೀಷನ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ? pollen grain ಒಂದೇ ಒಂದು pollen tube generate ಮಾಡುತ್ತೆ. ಅದಕ್ಕೆ monocyphinous condition. But Malvaceae ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ? Malvaceae ಅಂದ್ರೆ okra, hibiscus. Okay, in those plants ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ? Polycyphinous condition. ಅಂದ್ರೆ pollen grains will generate more than one pollen tube from the germ pores. Okay, so that is the example. ನೋಡಿ, ಇಲ್ಲಿ aperture of the germ pore ಇರುತ್ತೆ, ಒಂದೇ ಒಂದು generate ಆಗ್ತಾ ಇದೆ. That is monocyphinous in condition. But ಇಲ್ಲಿ ಏನಾಗ್ತಾ ಇದೆ? See, monocyphinous in condition. See, these are each pollen grain is developing into only one pollen tube from one pollen grain is monocyphinous in condition. See, Cucurbitaceae family ke example adu. This is the picture visualized for the given examples. Fine. The generative cell ಏನಾಗುತ್ತೆ will divide mitotically to form male gametes called sperms. We learned that generative cell ಒಳಗಡೆ 2 male cells ಆಗಿ develop ಆಗುತ್ತೆ, that is called as sperms. So these male gametes, sperms, are non-motile here. Non-motile means they are not having that capacity to move. They cannot move. That is why they are called as non-motile and amoeboid in structure. Amoeboid ಅಂದ್ರೆ ಏನು? They are shapeless. Perfect shape ಇಲ್ಲ ಅದಕ್ಕೆ. And they are also slightly unequal. 2 male gametes are not also equal to each other. ಒಂದೇ size ಇರುತ್ತೆ, ಒಂದೇ shape ಇರುತ್ತೆ ಅಂತಿಲ್ಲ. They are unequal, non-motile, not able to move, and also amoeboid in structure. Amoeboid and they are shapeless. They are not having any particular structure. So at this 3 cell stage, the pollen is called mature male gametophyte. 3 cell stage. Alva 2 cell stage next will develop into male gametes alva. આ stage ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ? Then completely the pollen, the gametophyte, male gametophyte is said to be matured in condition. Got it? See, this is the generative nucleus and this is the tube nucleus. ಇದೇನಾಗುತ್ತೆ? Pollen grain plus pollen tube. Pollen grain, pollen tube, The generative cell will be able to produce 2 sperm cells along with the tube nucleus. Tube nucleus will help in movement of the male sperms in the movement direction. It will take the sperms into a correct direction or help in the movement. Because we learned these male gametes are non-motile. Adage move agagalla. Adike tube nucleus in the help togondo it will move and also reach to the destination and able to fuse with the female nucleus. So the function of the pollen tube is to carry, as we learned, is to carry the non-motile sperms. Correct? So vegetative nucleus will enter first. Adike tube nucleus first hoggbeku. Illa andre ee male nucleus athwa sperms will never be able to move. Ade first move agutte and it is a vestigial structure. ಯಾಕಂದ್ರೆ ಅದಕ್ಕೇನು ಅಷ್ಟು ಪ್ರಾಮಿನೆಂಟ್ ಫಂಕ್ಷನ್ ಇಲ್ಲ, ಅದೇನು ಜಸ್ಟ್ ಮೂವ್ ಮಾಡಕ್ಕೆ, ಅದು ಸ್ವಲ್ಪ ಹೊತ್ತು ಮೇಲ್ ಸ್ಪಾಮ್ಸ್ ನ ಅದನ್ನ ಅದ್ರ ಜಾಗಕ್ಕೆ ತಲುಪಿಸಿದ ತಕ್ಷಣ ಇಟ್ ವಿಲ್ ಡಿಜನರೇಟ್. ಅದ್ರ ಫಂಕ್ಷನ್ ಮುಗೀತು ಅಂದ್ಬಿಟ್ಟು ಇಟ್ ವಿಲ್ ಜಸ್ಟ್ ಡಿಸ್ಟ್ರಾಯ್ ಇಟ್ ಸೆಲ್ಫ್. ಓಕೆ, ದೆನ್ ದಿಸ್ ಗ್ರೋತ್ ಆಫ್ ಪೋಲನ್ ಟ್ಯೂಬ್ ಸ್ಟಿಗ್ಮಾ ಇಂದ ಹಿಡಿದು ಓವರಿ ತನಕ ಬೆಳಿಬೇಕು ಅಂದ್ರೆ ಇಟ್ ಇಸ್ ಹ್ಯಾವಿಂಗ್ ಸಮ್ chemotropic in nature. ಏನ್ chemotropic? It will move towards the chemical substance. Chemical substance in the attract agi it will develop. Okay, and apically confined to the tip of the pollen tube. Adrindagi chemotropic in the nature irodrinda the pollen tube will be easily able to grow inside the carpel athwa female gametophyte. This is the whole picture again to have to say in conclusion. ಹೇಗೆ ಡೆವಲಪ್ ಆಗ್ತಾ ಇದೆ ಅಂತ summarize ಮಾಡ್ಬೋದು. See, you can understand this picture more easily. We are in this stage now. We finished all these stages. ಇಲ್ಲಿ generative cell ಆಗುತ್ತೆ, ನ್ಯೂಕ್ಲಿಯಸ್ ಬರುತ್ತೆ, ವೆಜಿಟೇಟಿವ್ ನ್ಯೂಕ್ಲಿಯಸ್, ಟ್ಯೂಬ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಇರುತ್ತೆ, ಸ್ಪರ್ಮ್ ಸೆಲ್ಸ್ ಇರುತ್ತೆ. ಅದೇ ಫಸ್ಟ್ ಇರ್ಬೇಕು. ನೀವು ಡ್ರಾ ಮಾಡಿದಾಗ್ಲೂ ಅಷ್ಟೇ ಟ್ಯೂಬ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಹಿಂದೆ ಹಾಕಿದ್ರೆ ದಟ್ ಇಸ್ ರಾಂಗ್, ದಟ್ಸ್ ನಾಟ್ ಹ್ಯಾಪನಿಂಗ್. ಟ್ಯೂಬ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಶುಡ್ ಬಿ ಇನ್ ದ ಫ್ರಂಟ್, ಸ್ಪರ್ಮ್ ಸೆಲ್ಸ್ ಹಿಂದೆ ಇರಬೇಕು. Then only it will go start developing the pollen tube till it reaches its purpose of destination. Okay, fine. ಈಗ pollen grain disadvantages ಏನು ಅಂತ ನೋಡೋಣ. What are the disadvantages of pollen grains now? Pollen grains of many species ಅಂದ್ರೆ animophilous species ಅಂದ್ರೆ wind pollination ಆಗುವಂತಹ plant species. And through wind, pollen grains will be transferred from one place to another. So, this type aliro plants alli, these pollen grains majority will cause allergies. Allergies cause madutte athwa bronchial afflictions andre related to the lungs, lung infection ಆಗಿರಬಹುದು ಅಥವಾ any ಉಸಿರಾಡೋ ತೊಂದರೆಗಳು ಆಗಿರಬಹುದು. ಇದೆಲ್ಲ ಆಗುತ್ತೆ pollen grains ಇಂದ. Okay. And there is one popular weed andre which is a waste plant, Parthenium anta. We have all seen in our locality, correct? Ee Parthenium plant, which is also called as carrot grass, India hege bandide, it has been imported to India with the improved wheat plant eno tarisbekare accidentally it has been entered India. So now this wheat has no function, but it is causing some allergies or disturbances in the people. So you can see this is the plant which has simply got some type of bronchial afflictions or allergies caused by it. So what are the simple advantages of pollen grains now? Pollen grains are rich in nutrients. Pollen grains are very rich in nutrients. And they are taken as tablets or syrups to improve the health. Nodi, idara supplements tara tagotare pollen grains because it is having more nutrient efficiency. So, tablet ರೀತಿಯಲ್ಲಿ ತಗೋಬಹುದು ಅಥವಾ syrups ರೀತಿಯಲ್ಲಾದ್ರೂ it is being marketed. Okay, these are the pollen products. And pollen consumption in the it is also being told that athletes ಅಥವಾ race ಮಾಡೋರು, horse racers and all, no, they will have more boost or ತುಂಬಾ energy ಬರುತ್ತೆ ಅವರಿಗೆ pollen grain supplements ತಗೊಂಡ್ರಿಂದ. But though there are so many illegal drugs being taken for them, ಅದೆಲ್ಲ before the competition they will check ಹೆಲ್ತ್ ಚೆಕ್ ಒಂದ್ಸತಿ ಫುಲ್ ಚೆಕ್ಅಪ್ ಮಾಡ್ತಾರೆ. ಸೊ ದಟ್ ಎನಿ ಇಲ್ಲಿಗಲ್ ಡ್ರಗ್ಸ್ ತಗೊಂಡಾಗ ದೇ ವಿಲ್ ಕಂಪ್ಲೀಟ್ಲಿ ಬ್ಯಾನ್ ದಟ್ ಪರ್ಸನ್ ಫ್ರಮ್ ದಟ್ ಒಲಿಂಪಿಯಾಡ್ ಆಗಿರ್ಬೋದು, ಕಾಂಪಿಟೇಷನ್ ಆಗಿರ್ಬೋದು. So there are some legal products being marketed that is fine to take. Okay, so this is also one of the advantage of bowling game. And see, these are all the sports. For sports kuda it will give enormous amount of energy, okay, for all the athletes. And viability andre adu yavaga functioning agirutte anta. Viability is the functioning, the period till which the pollen grains retain the ability to germinate. ಅಂದ್ರೆ ಇನ್ನ ಲೈವ್ ಆಗಿರುತ್ತೆ, ಇನ್ನ ಫುಲ್ ಡಿಜನರೇಟ್ ಅಥವಾ ಡಿಕಂಪೋಸ್ ಆಗದೆ ಇರಲ್ಲ. ದಟ್ ಕಂಡೀಷನ್ ಇಸ್ ಕಾಲ್ಡ್ ಎಸ್ ವಯಬಿಲಿಟಿ. ಸ್ಟಿಲ್ ಇಟ್ ಇಸ್ ಹ್ಯಾವಿಂಗ್ ದ ಕೆಪಾಸಿಟಿ ಟು germinate. The capacity to germinate is called as pollen viability. So ಅದು ಡಿಪೆಂಡ್ಸ್. ಇವಾಗ pollen grains ಡೆವಲಪ್ ಆದ ತಕ್ಷಣ 30 minutes ಅಷ್ಟೇ ಕೆಪಾಸಿಟಿ ಇರುತ್ತೆ. ಇನ್ ಪ್ಲಾಂಟ್ಸ್ ಲೈಕ್ ರೈಸ್, ವೀಟ್ ಇದೆಲ್ಲ 30 minutes ಅಲ್ಲಿ germinate ಆಯ್ತು ಅಂದ್ರೆ germinate, ಇಲ್ಲ ಅಂದ್ರೆ it is of waste, it will get degenerated. But in rosaceae, rose plants, legumes ಅಂದ್ರೆ beans, solanaceae, potato, ಈ plants ಎಲ್ಲ ಏನಾಗುತ್ತೆ several months together ಅದು ಹಂಗೆ ಇರುತ್ತೆ. ಅಂದ್ರೆ ಇದ್ರಲ್ಲಿ 30 minutes ಆದ್ರೆ ಎಷ್ಟು ಮಂತ್ಸ್ ಆದ್ರೂ ಯಾವಾಗ್ಲೇ ಆ ಪೋಲನ್ ಗ್ರೇನ್ ಯಾವ್ದಾದ್ರು ಪ್ಲಾಂಟ್ ಮೇಲೆ ಬಿದ್ರು ಅಥವಾ ಅದೇ ಪ್ಲಾಂಟ್ ಮೇಲೆ ಇದ್ರು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡೋ ಕೆಪಾಸಿಟಿ ಇನ್ನ ಇರುತ್ತೆ. ಬಟ್ ರೈಸ್, ವೀಟ್ ಇದ್ರಲ್ಲೆಲ್ಲ ಏನಾಗ್ಬಿಡುತ್ತೆ, ಕೆಪಾಸಿಟಿ ತುಂಬಾ ಕಡಿಮೆ. 30 minutes ಆದ್ಮೇಲೆ ಅದು ಫಿಮೇಲ್ ಗೆಮೆಟೋಫೈಟ್ ಮೇಲೆ ಬಿದ್ರು ಕೂಡ ಅಲ್ಲಿ ಪಾಲಿನೇಷನ್ ಆದ್ರೂ ಕೂಡ there will be no reproductive process happening. ಸ್ಟ್ರಾಬೆರಿ ಇದೆಲ್ಲ ರೋಸೇಸಿಯೆ ಫ್ಯಾಮಿಲಿ ಬರೋದು. ಸೊ ಇದರಲ್ಲೆಲ್ಲ ಜಾಸ್ತಿ ಸೆವೆರಲ್ ಮಂತ್ಸ್ ತನಕನೂ ಪಾಲಿನೇಷನ್ ಆಗಿ ರಿಪ್ರೊಡಕ್ಷನ್ ಆಗುವ ಕೆಪಾಸಿಟಿ ಇನ್ನ ಇರುತ್ತೆ. So we will conclude this by understanding the cryopreservation. ಇದನ್ನ pollen grains we said causal ಆಗಿ ಪ್ರಿಸರ್ವ್ ಮಾಡಬಹುದು ಅಂತ ಅಲ್ವಾ. ಅದೇ ತರ cryopreservation is also one of the scientific method to protect these or preserve these pollen grains. ಅಂದ್ರೆ ಇದನ್ನ pollen banks ಅಂತ ಕರಿತೀವಿ. pollen banks ಅಂದ್ರೆ ಈ scientific persons ಏನ್ ಮಾಡ್ತಾರೆ, they will take these pollen grains and preserve it in liquid nitrogen. Liquid nitrogen ಅನ್ನೋ chemical substance ತಗೊಂಡು gas ತಗೊಂಡು -196 degrees Celsius. Okay, -196 degrees Celsius ಅಲ್ಲಿ ಪ್ರಿಸರ್ವ್ ಮಾಡ್ತಾರೆ. ಅಂದ್ರೆ -196 degrees Celsius ಅಲ್ಲಿ ಇದ್ದಾಗ ಮಾತ್ರ ಪೋಲನ್ ಇನ್ನ ವಯಬಲ್ ಆಗಿರುತ್ತೆ, ಅಂದ್ರೆ ಸ್ಟಿಲ್ ಏಬಲ್ ಟು ರಿಪ್ರೊಡ್ಯೂಸ್. ಅಥವಾ ಅಲ್ಲಿ ಏನಾದ್ರೂ ಆಲ್ಟರೇಷನ್ಸ್ ಆದ್ರೆ ಅಗೇನ್ ಸ್ಟೋರೇಜ್ ಮಾಡೋದು ಕೂಡ ವೇಸ್ಟ್ ಆಗ್ಬಿಡುತ್ತೆ. ದಿಸ್ ಟೈಪ್ ಪ್ರಿಸರ್ವೇಷನ್ ಇಸ್ ಕಾಲ್ಡ್ as cryopreservation by using liquid nitrogen in very cold temperature, that is -196 degrees Celsius. Okay, let us understand with understanding all these topics, we will solve only few questions which are very important for CET and NEET. The first question comes up here, that is a typical angiospermic anther is bilobe and tetragonal, consisting of 2 microsporangium irutta, 3, 4 or only 1. This is the right answer. We learned that it is having 4 microsporangium. Next, what is the ploidy, monoploida, diploida of microspore tetrad? Tetrad ಯಾವ ಕಂಡೀಷನ್ ಇರುತ್ತೆ? Tetrad is haploid. MMC is still in diploid condition, but MMC ಯಾವಾಗ tetrad ಆಗುತ್ತಲ್ಲ meiosis 2 ಅಲ್ಲಿ ಅವಾಗ haploid ಆಗಿಬಿಡುತ್ತೆ. Which of the following weed has become a major cause of pollen allergy in India? Yes, which has been imported along with the beets? That is Parthenium. Intine is made of? Yeah, sporopollen. Yes? No, it is pectin and cellulose. Sporopollen yadu? It is in exine layer. Pollen viability for rice and wheat plant? 30 hours, 30 months, several months, 30 minutes, 30 seconds. Just now we learned that it is 30 minutes. So with that, we have finished the male gametophyte. Okay. In the chapter of sexual reproduction in flowering plants, one pre-fertilization, ಇವೆಂಟ್ ನ ಮುಗಿಸಿದೀವಿ, ಅಂದ್ರೆ ಮೇಲ್ ಗ್ಯಾಮೆಟೋಫೈಟ್ ಮುಗಿಸಿದೀವಿ. ಇನ್ ನೆಕ್ಸ್ಟ್ ಸೆಷನ್ ವಿ ವಿಲ್ ಮೂವ್ ಆನ್ ಟು ದ ಫೀಮೇಲ್ ಗ್ಯಾಮೆಟೋಫೈಟ್. ಅದ್ರ ಸ್ಟ್ರಕ್ಚರ್ ಏನು, ಇಂಟರ್ನಲ್ ಸ್ಟ್ರಕ್ಚರ್ ಏನು ಅಂತ ಸ್ಟಡಿ ಮಾಡೋಣ. So with that we will understand how exactly the reproduction process will happen. Okay, so hope you all understood. All the best. Thank you.